Welcome to Teolumen Insights, where we reveal the hidden forces that shape the way you live and show you how to use them to create real transformation and supercharge your health, mindset, and daily life. Here are your hosts, Emma Greenwood and Nathan Sinclaire.
Hello and welcome to Teolumen Insights. I'm Nathan.
And I'm Emma. And as always, we're so glad to have you with us today as we take a deep dive into a topic that honestly feels like it's straight out of science fiction, but it's something that's very much on the horizon of modern medicine.
Yeah, we're talking about nanorobotics in cancer treatment.
Exactly. And, you sent over some absolutely fascinating research articles on this, and I'm really excited to kind of unpack all this with you today and explore how these incredibly tiny machines, these nanorobots could really revolutionize how we treat cancer.
Absolutely. I think the sources you picked out really highlight the potential here. We've got studies looking at DNA-based nanorobots that are engineered to target tumors in mice. We've got breakthroughs in nanomaterials that can deliver drugs with pinpoint accuracy. And then we've also got kind of a broader overview of how bio-nanotechnology is impacting both the diagnosis and treatment of cancer.
Yeah, it's a lot to cover, but I think our mission today should be to really try to distill, the most critical insights from these sources about specifically nanorobotics in cancer treatment.
Right, and break it down in a way that everyone can understand.
Yeah.
We want to really understand how these nanorobots work. What are the advantages they might offer compared to, the current treatments that are out there?
Yeah.
And of course, what challenges still lie ahead before this becomes, a reality for patients.
Right, yeah. And I think maybe a good place to start would be with something that really jumped out at me when I was reading through this research, and that was these pH-responsive DNA nanorobots.
Yeah, these are really fascinating. So first of all, I think it's important to kind of adjust our mental picture when we hear the word nanorobots. We're not talking about, these little metallic robots like you see in the movies.
Right, yeah, no little Wallies running around.
Exactly. These are actually intricate structures that are built at the nanometer scale. I mean, one of the papers you sent, nanobots4.pdf, mentions a size of 90 by 60 nanometers. So to give you an idea of just how small that is, if a nanorobot were the size of a grain of sand, a human hair would be as wide as a football field.
Wow, that's incredibly tiny.
It is. And what's really remarkable is that these nanorobots can be designed to respond to specific cues in their environment. And one of the most interesting cues that they've been engineered to respond to is pH levels.
Okay, so why is pH so important when we're talking about cancer, and how does that tie into these nanorobots?
Well, it turns out that the microenvironment around a tumor is often more acidic than healthy tissue. So, healthy tissue will typically have a pH of around 7.4, but the area around a tumor can be significantly lower, often around 6.5.
So it's like the tumor has its own unique kind of chemical signature.
Exactly. And scientists have figured out a way to exploit this difference in pH to create nanorobots that can essentially target the acidic environment around a tumor while leaving healthy tissue untouched.
Okay, so how does that actually work? How do they build these nanorobots to be sensitive to pH?
Well, one of the papers you sent, S41565-024-01676.pdf, describes this really elegant mechanism called a DNA robotic switch. And essentially what this switch does is it allows the nanorobot to change its shape or its configuration in response to changes in pH.
Interesting.
Yeah, so in a normal pH environment, say like in healthy tissue, this switch is kind of in an off position. It's like a closed door. And the nanorobot is essentially inert. It's not really doing anything. But when it encounters the lower pH environment of a tumor, the switch flips to an on position. The door swings open, and this triggers a change in the nanorobot structure.
Okay, I'm starting to see how this could work. So what happens when that switch flips on? What does the nanorobot actually do?
Well, in this particular study, they designed the nanorobot so that when the switch flipped on, it would display these cytotoxic ligands.
Cytotoxic ligands, what are those?
They're essentially molecules that can kill cells. And in this case, they're specifically designed to kill cancer cells. So you can kind of think of it like the nanorobot has these little weapons that are hidden away when it's in a healthy environment, but then they're exposed when it reaches the acidic environment of a tumor.
So it's like a a targeted bomb that only goes off when it's in the right place.
Exactly. And the researchers found that this approach was really effective in killing cancer cells in vitro.
Okay, in vitro. So in the lab, but have they actually tested this in living organisms yet?
Yes, actually they did. They
Hmm.
tested this in a mouse model of cancer, and they found that these pH-responsive nanorobots were able to significantly reduce tumor growth compared to a control group.
Wow, that's really promising.
It is, and it really highlights the potential of this approach.
Yeah.
If we can design nanorobots that can selectively target and kill cancer cells while sparing healthy tissue, that could be a huge breakthrough in cancer treatment.
Yeah, absolutely. I mean, it could potentially mean, fewer side effects from treatment and maybe even better outcomes for patients.
Exactly. And it's not just about pH sensitivity either. Researchers are also exploring other ways to make these nanorobots even more targeted and effective.
Like what, for example?
Well, another paper you sent, nanobots4.pdf, talks about using biomarkers to target nanorobots to specific types of cancer cells.
Biomarkers. Okay, so remind me, what exactly is a biomarker?
So a biomarker is basically any molecule that can be used to identify a particular process or condition in the body. And in the context of cancer, there are certain biomarkers that are specifically expressed on cancer cells or in the tumor microenvironment.
Right, so it's like a flag that says, hey, I'm a cancer cell, or hey, this is a tumor.
Exactly. And one biomarker that's been studied quite a bit in relation to nanorobotics is a protein called nucleolin.
Nucleolin. Okay, and what's special about nucleolin?
Well, it turns out that nucleolin is found in high concentrations on the surface of tumor endothelial cells.
Endothelial cells, what are those?
So those are the cells that line the blood vessels. And in the case of tumors, these blood vessels are really important because they supply the tumor with the nutrients and oxygen it needs to grow. So if you could target those blood vessels, you can essentially cut off the tumor supply lines.
Oh, that makes sense.
Right, and that's where nucleolin comes in because it's a really good target for nanorobots because it's so highly expressed on tumor blood vessels, but not on healthy blood vessels.
So you're basically giving the nanorobots an address to go to. You're saying, look for this specific protein, and you'll find the tumor's blood supply.
Exactly, and one way to do that is to use something called DNA aptamers.
DNA aptamers. Okay, so what are those, and how do they work?
So aptamers are basically short, single-stranded pieces of DNA or RNA that can be designed to bind to very specific target molecules.
So they're kind of like little molecular homing missiles?
Yeah, that's a great analogy.
Mm-hmm.
And in the case of nucleolin, researchers have been able to create aptamers that bind to it with very high affinity.
So you could attach those aptamers to the nanorobots and essentially program them to seek out and attach to any cells that are expressing nucleolin, which in this case would be those tumor blood vessels.
Exactly. And that's exactly what they did in the study you sent. They attached nucleolin targeting aptamers to their DNA nanorobots, and then they injected those nanorobots into mice with tumors.
And what happened?
Well, they found that the nanorobots were able to successfully target the tumor blood vessels and deliver their payload, which in this case was an enzyme called thrombin.
Thrombin. Okay, and why thrombin?
Well, thrombin is a key player in the blood clotting process.
Right, so by delivering thrombin to the tumor's blood vessels, you're essentially causing blood clots to form, which would block the flow of blood to the tumor and starve it.
Exactly, and that's exactly what they saw in the study. The nanorobots were able to significantly reduce blood flow to the tumors, and that led to a reduction in tumor growth.
Wow, so it's like a double whammy. You're not only targeting the tumor directly with these cytotoxic ligands, but you're also cutting off its blood supply.
Exactly, and it's a really good example of how researchers are starting to combine different approaches to make these nanorobots even more effective.
And one of the things that I find really exciting about this whole field of nanorobotics is that it's not just about treatment, it's also about diagnosis, right?
Absolutely. I mean, one of the papers you sent, Nanomaterials for Drug Delivery in Cancer Therapy.pdf, talks about how nanomaterials can be used to create incredibly sensitive diagnostic tools.
Right.
And one example they give is RNA-DNA nanobiosensors.
Nanobiosensors.
Yeah.
Okay, so what exactly are those?
Well, basically, they're nanoscale devices that can detect and measure specific molecules or biological processes in the body. And in the context of cancer, they can be used to detect things like tumor markers or even the presence of cancer cells themselves.
So it's like having a tiny little lab inside your body that can monitor for signs of cancer.
Exactly. And these nanobiosensors can be incredibly sensitive. I mean, they can detect molecules at concentrations that are far lower than what's possible with traditional diagnostic techniques.
Wow, that's amazing. And how do they actually work? How do they detect these molecules?
Well, there are a lot of different ways to design nanobiosensors, but one common approach is to use aptamers.
Those homing metals again?
Exactly. So you can design an aptamer that specifically binds to a particular tumor marker or cancer cell, and then you can attach that aptamer to a nanoparticle. And when the aptamer binds to its target, it triggers a change in the nanoparticle that can be detected using various techniques.
Okay, so for example, you could have a nanoparticle that fluoresces when its aptamer binds to a cancer cell. And then you could use a special microscope to see those fluorescent nanoparticles, and that would tell you where the cancer cells are.
Exactly, and the paper you sent mentions a number of different fluorescent dyes that can be used for this purpose, like FITC CC55, CSI3, and AF647.
So it's like tagging the cancer cells with these little glowing beacons?
Exactly, and that can be really useful for both diagnosis and for monitoring treatment response.
Right, so you could see if the treatment is actually working by tracking how many of those glowing beacons are still present after treatment?
Precisely, and Nanobots 5.pdf mentions another really cool technique called multimodal imaging.
Multimodal imaging. Okay, so what is that?
So basically, it's the idea of combining different imaging techniques to get a more complete picture of what's going on in the body.
So instead of just looking at one type of image, say like an X-ray, you could combine that with other types of images like MRI or PT scans.
Exactly, and by combining different imaging techniques, you can get a much more detailed and informative view of the tumor and its surroundings.
That makes sense. And how does that tie into nanorobotics?
Well, you can design nanoparticles that can be detected by multiple imaging modalities. So, for example, you could have a nanoparticle that is both fluorescent and radioactive.
Okay, so you could use fluorescence microscopy to see where the nanoparticle is located, and then you could use PT scanning to see how active the tumor is in that location.
Exactly. And that kind of information can be really valuable for both diagnosis and treatment planning.
Absolutely. So we've talked about how nanorobots can be used to target tumors and how they can be used to diagnose cancer. But what about the actual treatment itself? How do these nanorobots actually kill cancer cells?
Well, we've already talked about one mechanism, which is using thrombin to cut off the tumor's blood supply. But there are other mechanisms as well. One that's mentioned in several of the papers you sent is apoptosis.
Apoptosis. Okay, I've heard of that before, but remind me what it is.
So apoptosis is basically programmed cell death. It's a process that happens naturally in the body to get rid of old or damaged cells.
Right, so it's like the cell's self-destruct button.
Exactly. And there are certain signals that can trigger apoptosis in cells. And one way that nanorobots can kill cancer cells is by delivering those signals directly to the cancer cells.
Okay, so how do they do that?
Well, one approach is to use those DNA robotic switches that we talked about earlier. So you can design a switch that is activated by something that is specific to cancer cells, like a particular protein or enzyme.
Okay, so when the nanorobot encounters a cancer cell, the switch flips on, and that triggers the release of a signal that tells the cancer cell to self-destruct.
Exactly. And the paper you sent, S415650240167764.pdf, describes a switch that is activated by the acidic environment of a tumor.
Right, so it's kind of a two-pronged attack. The nanorobot is attracted to the acidic environment of the tumor, and then once it's there, it's activated to release this apoptosis-inducing signal.
Exactly, and there are other approaches as well. Another way to trigger apoptosis is to use nanoparticles that can generate reactive oxygen species.
Reactive oxygen species, what are those?
So they're basically molecules that can damage DNA and other cellular components. And when a cell is exposed to too many reactive oxygen species, it can trigger apoptosis.
Okay, so you could have a nanorobot that releases these reactive oxygen species directly into the tumor.
Exactly. Or you could have a nanorobot that absorbs light and then uses that energy to generate reactive oxygen species.
Interesting. And is that something that's being actively researched?
Yes, definitely. There are a lot of different groups working on developing nanorobots that can generate reactive oxygen species for cancer treatment.
And how does this approach compare to traditional cancer therapies like chemotherapy and radiation?
Well, one of the biggest advantages of using nanorobotics for cancer treatment is that it has the potential to be much more targeted than traditional therapies.
Right, because with chemo and radiation, you're basically blasting the entire body with these toxic substances or radiation, which can cause a lot of side effects.
Exactly. And with nanorobotics, the idea is to deliver the treatment directly to the tumor so that you minimize damage to healthy tissue.
And that could potentially mean fewer side effects and a better quality of life for patients.
Exactly. And there's also the potential for nanorobotics to be more effective than traditional therapies.
How so?
Well, because you're delivering the treatment directly to the tumor, you can achieve much higher concentrations of the drug at the tumor site.
So you could potentially kill more cancer cells with a lower dose of the drug.
Exactly. And that could lead to better treatment outcomes.
This is all incredibly promising, but I imagine there are still some challenges to overcome before this technology becomes widely available.
Absolutely. I mean, this is still a relatively new field, and there's a lot more research that needs to be done. But one of the biggest challenges is scalability.
Scalability, what do you mean by that?
Well, it's one thing to create these nanorobots in the lab, but it's another thing entirely to manufacture them on a large enough scale to treat patients.
Right, because you need to be able to produce these nanorobots in a way that is both cost-effective and reproducible.
Exactly. And that's not easy to do with nanoscale materials.
So what are some of the approaches that researchers are exploring to address this challenge?
Well, one approach is to use self-assembly.
Self-assembly? What is that?
So self-assembly is basically the process of designing molecules that can spontaneously assemble into larger structures.
Okay, so you could design these molecules so that they would automatically form into nanorobots when you mix them together.
Exactly. And that could potentially make it much easier and cheaper to manufacture nanorobots on a large scale.
Interesting. Are there any other challenges that researchers are facing?
Yeah, another big challenge is the immune system.
Ah, right, because the immune system is designed to attack anything that it perceives as foreign, which would include these nanorobots.
Exactly. And so researchers are working on ways to either disguise the nanorobots from the immune system or to suppress the immune response.
How are they doing that?
Well, one approach is to coat the nanorobots with materials that are biocompatible.
Biocompatible, meaning that they don't trigger an immune response.
Exactly. And another approach is to use nanorobots that are made from materials that are naturally found in the body, like DNA or lipids.
So it's like you're making the nanorobots look like they belong there so that the immune system doesn't attack them.
Exactly. And there are other approaches as well. Some researchers are even exploring the possibility of using bacteria as nanorobots.
Bacteria.
Yeah, so certain types of bacteria have the ability to target tumors, and they can also be engineered to deliver therapeutic payloads. So it's possible that we could one day use bacteria as a kind of living nanorobot for cancer treatment.
Wow, that's fascinating. It sounds like there are a lot of really exciting possibilities in this field.
Definitely. This is a field that is moving very quickly, and I think we're going to see some really amazing advances in the years to come.
Absolutely. Well, this has been an incredible deep dive into the world of nanorobotics in cancer treatment. It's clear that this is a field with enormous potential, and I'm really excited to see what the future holds.
Me too. And before we wrap up, I just wanted to leave our listeners with a thought-provoking question.
Okay, I'm all ears.
So we've talked about how nanorobotics could revolutionize cancer treatment. But what other diseases or medical conditions do you think could potentially be treated using this technology?
That's a great question. I mean, if we can design nanorobots that can target specific cells and deliver therapeutic payloads, then the possibilities are really endless, right?
Exactly. I mean, imagine nanorobots that could deliver drugs to the brain to treat Alzheimer's disease, or nanorobots that could repair damaged heart tissue after a heart attack.
Yeah, or nanorobots that could target and destroy viruses like HIV or the flu.
Exactly. The potential is truly mind-boggling.
Absolutely. Thank you so much for joining me today for this deep dive into nanorobotics and cancer treatment.
My pleasure.
And for our listeners, if you enjoyed this episode, please be sure to like and subscribe to our channel for more deep dives into the world of science and technology.
Till next time, this is Nathan
And Emma.
signing off from Teolumen Insights.
Goodbye.
Goodbye.